I think I'm going to get started. I guess.
Okay, great.
Most people are probably here. So, all right. So I'm going to put my screen so I can see your slides. Well, great. All right. So it's my pleasure today to introduce Dr. Shen Luang to the genetics seminar. Shen obtained his PhD in 1991 at Harvard University in the Department of Cell and Developmental Biology. He followed up with a postdoc at Harvard University again. And in 1994, he was appointed assistant professor in the Department of Plant and Microbial Biology at UC-Berkley. And he quickly moved up the ranks to become full professor in 2004. And he is still in this department. And in fact, since I think last year, if I'm correct, you are the chair of this department. So you're, I'm sure, very busy. So thank you very much for spending some time to talk about your research today. Shen Luang's laboratory studies how overall plants perceive and respond to a diverse range of environmental signals. And his research is focused in particular on calcium signaling, including the coding of calcium signature and ion channels, calcium channels, and decoding of calcium signature, and everything that happens downstream in terms of how all the biochemical process that have happened downstream, and basically, again, how controlling how plants respond to the environment, and looking at different parts of the plant cells from plasma membrane, vacuole, chloroplast, and we were just talking about nuclei. So Shen is very active also, not only in research, but also as a member of the plant science community. As I said, he's currently serving as the chair of the Plant and Microbial Biology Department at UC-Berkley. But for instance, he's also the founding editor of a very popular journal, Molecular Plant, that from 2008 to now. He received also many honors, including the Alexander von Humboldt Research Award in 2008, awards from the ASPB, American Society for Plant Biology, and in 2012, he was named a AAAS Fellow. So highly recognized as a great member of our community. So without further ado, please join me in welcoming Dr. Luang to the genetics seminar.
Okay, thank you, Jean-Michel, for the invitation and the opportunity for me to share with you some of the work in my lab today. So we know that calcium is a very special mineral. It's not only a nutrient, but also a signal. When it serves as a signal, it actually can regulate the homeostasis of other minerals in plants. So this is the kind of main topic of what I'm going to share with you today. Along the way, I'm also going to give a couple simple examples how we deal with, calcium coding and decoding network. Okay, so plant need at least four things to grow, right? While CO2 and sunlight usually are plenty, water and the minerals are limiting factors very often in nature, right? So in nature setting, and also that's why you need a lot of fertilizers and irrigation for modern agriculture. And just to focus on the fertilizer, it's predicted that this half century, this half of the century, really the yield increase of crop production is going to heavily depends on fertilizer use. It's predicted that 50% yield increase is going to be on the fertilizer use. So in other words, without fertilizer, we're going to cut the yield in half. Then everybody become hungry, right? And this prediction lead to these two curves. The population growth and fertilizer use is going to move up hand in hand, right? And very closely. So that is not sustainable because we know many fertilizers come from natural resources that are being depleted. For example, phosphorus, rock, and potash, they can only be used probably for a couple hundred years. Some many years down the road, but after this couple hundred years, then nothing will be left, right? So that's not sustainable if we keep using a lot of fertilizer. And another important factor for this sustainability concern is really environmental impact, right? So we know that a lot of fertilizer runoff into our freshwater resource causing eutrophication of our lakes and rivers and really damaging our ecosystem. So this heavy fertilizer use is really not sustainable. So a new green revolution in agriculture really involves reduction of chemical input, including fertilizer. Of course, probably fertilizer is the largest chemical input in agriculture. And this green revolution, of course, is considered important for support this sustainable agriculture, which is obviously the center to support our society, environment, and economy. So to reduce the fertilizer use specifically, we need to, I think the general strategy is really to improve nutrient use efficiency of plant. So the NUE, or nutrient use efficiency, is determined by many factors, including uptake of the different minerals, distribution to different organs, and storage of the nutrient, reallocation during development, for example, from vegetative growth to reproductive growth development. Then you need reallocation of the or distribution of the nutrient to the different organs, like flower, fruit, and the seed, right? So for perennial plant, then at the end of the growing season, you need to kind of redistribute, recycle the nutrient from the falling leaves, to back to the storage part. So all these processes entail membrane transport that involve a lot of transporters and pumps, and that very often hold the key or become limiting factors for nutrient utilization. And if we look at the single cell level, so membrane transporter not only move nutrient in and out of cell, but also distribute nutrient within each cell among different organelles, right? Because every organelle has its own membrane that border or serve as a boundary. So the concept of nutrient homeostasis is really to balance the demand and the supply of all minerals at different levels. Ideally, all the organelles in the cell and all the cells in a single plant receive a perfect balance of nutrient. So that's what, the ideal situation. That it's not very often possible, but plants are constantly trying to achieve that. Okay, how do they do that? Basically, what they do is they would channel all the signals, right? They'll monitor the supply of the nutrient outside of the cell and the demand of the nutrient inside the cell. They use those as signals. They integrate those signals and channel those signals to the regulation of the activity of those transporters in the membranes. In a way that transporters in the membrane really like doors and windows, right, in our house. It's not always open or closed. That way, if you always open or always close, then it's loss of function. The door or window is a loss of function, right? So it has to be open or closed upon demand, right? That is what the cells are doing. They channel or integrate those signals to the control of the activity of those doors and windows in the membrane, so that they can decide when to transport and what to transport, where they want to transport all those different nutrients. Okay, if we summarize that, into a kind of conceptual framework is really the homeostasis or neutron sensing and homeostasis are really controlled by the integration of the two networks. Okay, the signaling network channel those supply and the demand signals to regulate the activity of this transport network. I say it's transport network because this network consists of literally thousands of different transporters in plant, right? So in the signaling network, of course, we have calcium serve as a signal messenger, not only in plant, but in all the eukaryotic systems, right? In response to many different signals, including environmental cues, and that, of course, further consists of biotic or abiotic signals, and developmental cues or internal signals that include hormones or other developmental signals. So then the first thing very often plant cells do is to change the calcium levels in the cell. And that calcium signal then is very often required and sufficient. That's how you define signal messenger, right? So not only required, but also sufficient to induce downstream physiological responses. Now, the big question mark here is really how calcium, a simple canine, translates so many different signals into distinct physiological responses, right? We know that plant respond to temperature change differently from pathogen response, right? But yet in all the cases, calcium is signal messenger. How does calcium specify, or where's the specificity is encoded in this calcium change? Studies in the past half century, 50, 60 years, active research, especially in animal and fungal systems, and of course, the plant research also contribute to that, is establishment of a calcium signature concept. Basically, that is to say that different signals formulate or produce a different calcium code or calcium signatures. And that calcium signature is really a four-dimensional parameter, right? So it consists of where the calcium signal in the cell localized, let's say it's in the nucleus or close to plasma membrane, right? So where, that's a 3D. And then on top of that, you have a time course. Let's say the signal comes to start from here, and then along the time course, the temporal pattern will change. This blue pattern, as we can tell, clearly differ from the orange pattern, right? And cells then should also tell the difference by decoding mechanisms that distinguish the kind of signature, and then formulate a specific response through decoding mechanism. And that decoding mechanism start with the calcium sensors. Often those sensors are calcium binding proteins, such as chemodulin, CDPK, CBL, etc. And then by binding calcium, they change their conformation. The conformational change lead to interaction with different targets, regulating their activity, and those targets can be, enzymes involved in signal transduction and transcription factor, regulate gene expression, transporters, and affecting transport. You name it. There's so many biochemical reactions. It can be downstream target for calcium sensors. Now, all these different reactions or responses will be literally calculated by the cell into, and then finally, formulate a specific response, depending what signals are particularly there present at a particular time. Okay, so this in a nutshell is really summarize how calcium-dependent signaling work, not only in plant, but also in other eukaryotic systems, right? How specific signals get channeled through coding and decoding of a calcium signal into specific response. My lab, as Jean-Michel mentioned, has been working on both calcium coding and decoding mechanisms through the years, more than 20 years now. And I'm going to give you just a very quickly a couple examples of how we study calcium coding mechanisms, and then focus most of my time on this decoding mechanism, especially the CBL, CIPK signaling network, and then how that network regulates mineral nutrient homeostasis. So to start, I want to just show you a couple examples how we work on this coding mechanism. So we know for a long time that in response to pathogenic bacteria, plant cells need calcium as a signal messenger. Okay, so if you block a calcium entry by calcium channel blockers, or just remove external calcium, then the pathogenic the immunity or immune response cannot happen, right? But the channels, the calcium channels are responsible for pathogen-induced calcium entry has not been understood until recently. We identify a pair of CNGC channels that actually serve as an important component in this pathogen response calcium signaling. Before pathogen attack, then this CNGC tetramers and tetramers are gated closed by chemodulin binding. And in response to pathogen patterns, these pattern receptors activate BIK1, which is a kinase, protein kinase, that then interact with the CNGC channels and the phosphorylated channel to open the channel so that calcium get influx, get into the cell to serve as a signal messenger to trigger ROS burst and other immune response. So this is an important part of the pattern triggered immunity in plant. Another example is reproduction in plant. We know that from pollen germination, pollen tube growth and guidance, all the way to arrive at the ovule, and then pollen tube interact with the synergy cell to trigger cell death and then sperm release, followed by fertilization. So each step of the way involves calcium as an important signal. And this calcium pattern very often show a oscillating pattern. Okay, so I'm now showing you how we are studying this process. So by basically giving you a sense of real-time dynamics of the calcium from the, this is a pollen grain. I'll show you. You see this focal point of calcium. This is where the pollen tube is going to emerge. Okay, so now it grows, and then the focal point of the calcium flash, right, will actually follow the tip of the pollen tube. So that we call it tip calcium gradient and also oscillation. You see the flashy. Now, it's not always there. It's just a flash, flash, flash. That basically, if you record the up and down, you see this kind of a oscillating pattern. Okay, so the oscillating pattern, anything oscillating, it has to be regulated by this kind of auto-regulatory feedback mechanism, right? So another example is circadian rhythm in plant and in other eukaryotic systems. So they're all controlled by auto-regulatory feedback transcription factors, right? But in this case, we discovered that another pair of CNGCs, there are 20 of those in Arabidopsis. Another pair of CNGCs interact with each other and regulated by interacting with chemodulin and formulating this autoregulatory feedback. So how does this work? So under low calcium, right? So the chemodulin will be empty, will not bind the calcium because the calcium is low. So that we call the apo chemodulin will actually interact with the tetrameric CNGCs and activate the channel activity. Okay, now the calcium get into the cell and the level of calcium start to go up, right? So go up until the peak. Let's say reach this peak, and then the chemodulin will bind the calcium, and that changes the conformation of chemodulin that trigger also the dissociation of the chemodulin from the channel and shutting down the channel, right? At the peak, the channel is closed. Then that would induce a downturn of intracellular calcium. And then when calcium reaches this bottom part, right, bottom level, and then what happens is that the chemodulin will get rid of calcium, cannot bind to calcium anymore, then become a calcium empty form, right? This apo called apochemodulin will again interact with the tetramer channel and activate the channel. Then the intracellular calcium again goes up until the peak, and then the chemodulin will bind the calcium, get dissociated from the channel, and start another cycle of activation and inactivation. So how this up and down is caused by, it's entirely based on the calcium level. In other words, it's a calcium dependent calcium level regulation, right? Through this channel up and down, open and close by a calcium sensor chemodulin in this case. Anyway, so we also identify another channel that's something we're writing a paper on. Another channel is also involved in this process, and how this work together is a big question, and a lot of people are interested in that. Okay, so this is how the calcium oscillation is happening at the pollen tube tip, or even at this beginning of germination, pollen tube germination, right? So when the pollen tube keep growing with the guidance of this oscillation, right? They grow, and until they arrive to the ovule of the flower, the pollen tube now here, and then the synergid will respond with their own calcium oscillation, right? And how this works is a mystery until recently. We published another paper, it's just 2 months ago actually, identifying basically another channel. It's an NTA or it's an MLO type family protein. It turned out that this whole MLO family, which stands for a mildew locus O, right? So it's involved in a mildew fungal tolerance, and also in other processes. So there's a big family of proteins. So one of the family member is right here. So the pollen tube actually produce rough peptide, right? And then released to synergid that is recognized by the surface receptor Pheronia LRE. And this co-receptors then recruit this NTA calcium channel to mediate the calcium entry into synergid. And when the calcium level is going up, they bind the chemodulin, and the chemodulin then interact with the channel, shut down the channel. So that produces, again, the oscillating pattern of the synergid. Let me show you how this look like at using single cell calcium imaging. You see the synergid cell is like a heartbeat, right? So go up and down, generating this oscillating pattern. in response to ROF4. And the mutant, all this mutant, lacking these receptors or channels, this heartbeat basically either stops or dramatically reduced. So this is just to show how we utilize this pretty nice technology, like single cell calcium imaging, for example, to really monitoring what's going on in real time and understand using genetics, how those different components are involved to encode that different calcium pattern in the cell. Okay, so with that, I just to show you some examples how we deal with calcium coding mechanism by identifying different channels and transporters and how they're regulated or how their activity are regulated to formulate those up and down patterns, right? Those up and down patterns, oscillating patterns, is due to the opening and closing of the channels, right? So now I'm going to use the rest of the time to talk about a particular CBL, CIPK, calcium decoding network, and how that regulates mineral nutrient homeostasis in plant. Okay, so that actually went back all the way to the time when I was a postdoc in Harvard Chemistry Department. I was studying a calcium-activated molecular switch called a calcineurin, which is a chemodulin calcium-dependent protein phosphatase. And the catalytic subunit A need to interact with regulator subunit B and chemodulin in response to calcium to form this trimeric active enzyme. Okay, so without calcium, this enzyme is pretty much low activity or inactive. In response to calcium, the activity, the trimeric enzyme complex occur and then become active and start to regulate, different processes by dephosphorylating proteins, right? The phosphatases dephosphorylate proteins. So this is how typically calcium sensing or calcium decoding steps, the first step really work, right? So you start with a calcium signature elevation, and then the calcium sensors bind to calcium and cause enzymatic activity change or conformation change of some proteins, and eventually lead to activity change in the cell. This is a typical mechanism or paradigm, right? So I was looking for something similar in plant, and long story short, I end up with cloning of a protein from Arabidopsis that's similar to or related to calcineurin B. So we call it calcineurin B-like proteins or CBL, right? So at that time, because if you compare the calcineurin B protein sequence between animal and yeast, they're highly conserved. They're like 70% identical. But CBL from plant is only like a 35% identical. So it doesn't seem to be a real homolog. But another very important factor that would decide whether this CBL is a true homolog of calcineurin B or not is really the target, right? So CNB regulates CNA, which is a protein phosphatase. If we identify a target as a protein phosphatase from plant, then you can say, oh, I find it a homolog, right? But if something else, then we have to redefine the function of CBL. So we identify the CBL interacting proteins, and all of those turned out to be a family of protein kinases. That's how the CIPK was born, and we call CIPK, of course, CBL interacting protein kinases. And it turned out to be a big family of protein kinases, kind of unique in plant. We call that also SNRK3 subfamily because it's related to SNF1 in yeast or AMPK in animals, right? So plant have 3 subfamily of SNRKs. We call it SNRK1, SNRK2, SNRK3. So CIPKs are considered or also called SNRK3 type protein kinases. So all the SNRKs have a very similar kinase domain, but the regulatory domain are very different. For CIPKs, so they have this red box in the C-terminal have that interact with CBLs, right? So this finding really redefined the kind of calcium signaling mechanism away from animal and fungi. In animal and fungi, at least in the case of calcineurin, we know that calcineurin B and calmodulin in response to calcium interact with protein phosphatase activity. But in plant, a calcineurin B-like protein interact with a bunch of protein kinases and activate kinase activity to pass the signal along. So phosphatase versus kinase, exactly kind of opposite biochemical entity, right? So this is very different and is therefore I think it's more interesting than finding a homolog, right? So in plant, actually, this area is blooming quite well in the past 20 years after the discovery of CBL-CIPK interaction network. So in plant, like in Arabidopsis, at least 10 CBLs are present, are encoded by genome, 26 CIPKs. So each CBLs, we realize, interact with usually multiple CIPKs. And each CIPK interact with at least one, very often multiple CBLs. So some CBLs share common CIPK target, and some CIPKs share CBL regulatory subunit. So this kind of specificity and overlap in protein-protein interaction would reflect functional redundancy and specificity of this interacting network. Now, how to turn this protein-protein interaction network into a functional network? Then we need to look for the signals and the calcium signatures that are decoded by the CBL CIPK, right? That are upstream signals. And then we also need to identify the downstream target or substrate for those kinases to piece together a whole pathway, right? So using different functional assays, so we did just that in the past 20 years also. I'm just going to give you a couple examples how this CBL-CIPK regulate potassium homeostasis in plant. So starting with a study on CBL1 and 9, which are highly identical, highly similar, right? More than 90% amino acid identity. And so with that kind of identity or similarity, we can predict that they probably function redundant. And also because their function is determined by interacting CIPKs. So we need to find what CIPKs they interact with, and we realize that most of the CIPK target are shared. That's predicted, expected, right? But we also find that some new or specific CIPK interactors. Sure enough, the CBL1 and 9 single mutant also have some phenotype. That means they have specific functions going through specific specific CIPK target, right? But I'm going to show you just the common function in the context of potassium homeostasis. Our study shows that actually calcium signal in response to low potassium status in the medium or in the soil trigger activation of CBL1 and 9 that get associated with the plasma membrane recruit the CIPK23, and that in turn interact physically with the AKT1 and phosphorylate this potassium channel, and which is responsible for potassium uptake. And AKT1 functioning potassium uptake is actually discovered by Edgar Spalding and Mike Sussman in your school. And so for a long time that people knew, for a half century, people knew that if you start a plant with a low potassium, and then a few days later, you compare the starved plant with the control well-fed plant, you do a potassium uptake assay. And the starved plant uptake potassium much faster than the control group, right? And that makes sense physiologically, because when you're starved, you eat faster, right? So that, but molecular mechanism underlying this low potassium enhanced potassium uptake process has not been known until our finding of CBL, CIPK regulation of AKT1. Okay, so basically findings in other labs show that low potassium can trigger ROS and also calcium signal. And that downstream that is really CBL, CIPK complex targeted to plasma membrane. By the way, this one, CBL, is targeted to plasma membrane by myristoylation, lipid modification. Okay, so that target there. And then CIPK then interact and phosphorylate AKT1, open the channel. So potassium can flow into the cell very quickly. Now, if CBL CIPK is a kinase, right? Kinase phosphorylation activates the channel, then you would expect some negative regulators or phosphatases might dephosphorylate the channel and serve as a negative regulator to reset the system, right? Signaling, it's not only about activation, but also inactivation, right? It's always on and off. Full cycle involve activation and inactivation. So in this case, we identify a PP2C type of protein phosphatase that interact with not only AKT1, but also with the CIPK23, form this physical triangle, actually. So the AKT1 activity then is fully based on, depends on how PP2C and CIPK are activated. Or in other words, the ratio of negative regulator versus active regulator determine the activity of AKT1. Here's an example. If you just include the CBL, CIPK together co-express with AKT1, the channel activity is huge, right? Now, on top of that, you co-express PP2C, the channel get much smaller. You can play around with the channel activity by introducing different ratio of negative regulator and positive regulator. You can pretty much control how much the channel output is. So this, I want to just really point out that this is a part of a bigger concept in biological regulation, right? So it's often quantitative regulation, right? So it all depends, the final output is really depends on the ratio of active transporter and negative transporter. So this applies to many signal transduction processes. Another important concept is that if you want to do things together, you need to come together physically. So this kind of a protein-protein interaction, physical interaction is really determine the functional specificity in this case, because if you want to do participate in the same process, you need to be in the same spot. Okay. Now, in summary, basically, I just, without showing all the detailed data, I just want to summarize this part of the talk. It's really this AKT1 Okay, so and CBL, CIPK, PP2C system form a signal transduction network pathway that respond to potassium status in the soil. Okay, when the soil potassium is high, in this case, if you add 1 millimolar potassium to the growth media, AKT1 is entirely dispensable. You don't need AKT1 or CBL, CIPK. So all those mutants grow just fine. If you have, 1 millimolar potassium is not that high. Many of our fertilized soil can have that. Okay, so, but most of natural soil has sub-millimolar potassium level, and therefore this system becomes very important. So in response to low potassium in natural soil or in some poor, farming land, then the CBL, the calcium-dependent CBL-CIPK system will be turned on, and that physically interact with and phosphorylate AKT1 channel, open the channel activity. And then the dephosphorylation, of course, set the system back and form this signaling cycle in response to potassium status in the soil. So all this nutrient status control the homeostasis, and this is a big area we call this nutrient sensing process. Now, if we put this pathway into a bigger picture, how potassium homeostasis works in plant, I gave you this picture. We started with the root hair. Okay, this is a root hair cell, let's say. There are two important potassium transporters in charge of potassium uptake. AKT1, I just mentioned, and HAC5, which is believed to be a proton potassium symporter. Okay, so both these two transporters are actually activated by CBL CIPK pathway I just mentioned. Okay, so without telling you all the detailed research. Anyway, the potassium get into the root hair and then transfer to different cells, and then all the way to the xylem through a outward potassium channel called SCORE, discovered by Syntenix's lab in France and Montpellier. And this potassium then, of course, go up to the chute through the transpiration stream, right? Go all the way up. And then I'm just using a mesophyll cell as example how this different cells handle potassium distribution, right? So of course, potassium can then get into this mesophyll cell through inward potassium channel, and then extra potassium will be stored into the vacuole through the NHS type of antiporter, proton antiporter. And then if the soil has a lot of potassium, then it's just keep going and then keep storing in the vacuole. But when the soil potassium get low, then this vacuolar potassium will flow out through the TPK, two-pore potassium channels, into the cytoplasm to support metabolism. So in response to low potassium, two important mechanisms take place to cope with low nutrient status. One is activation of these two transporters so that more doors and windows are open, wide open, and more potassium will flow in. I just showed you earlier. And then another important mechanism is to remobilize stored potassium in the vacuole to support metabolic processes in the cytoplasm. Okay, so I already shown you an example how this one mechanism is regulated by CBL-CIPK pathway, and I'm going to show you some real data to show you that in fact the second mechanism, the vacuolar potassium remobilization through TPKs, are also regulated by CBL-CIPK, but another different CBL-CIPK pathway. Remember, there's many CBL CIPKs, right? So let me show you some data here. So unlike CBL 1 and 9, I just shown you earlier that localized to plasma membrane and regulated those plasma channels, CBL 2 and 3 are also very similar proteins, and they are both targeted to vacuolar membrane. Okay, if it's not that clear, you just believe me, they're associated with the vacuole. And if you do genetics, single mutant, because they're very similar, single mutant do not show any visible phenotype. But when you have a double mutant, they start to show a smaller plant and all those nutrient deficiency or imbalance kind of phenotype, burning tip, leaf, and things like that. This phenotype, nutrient phenotype start to show. Now, is this phenotype related to potassium remobilization from vacuole, as CBL 2 and 3 are targeted to vacuole, right? So in the context of potassium, I want to show you two assays. So how does potassium remobilization from vacuole function in plant growth? One very important process is seed germination, right? We know that you can germinate your Y-type seed in pure water, no problem, right? Because germination and establishment of seedling can utilize the stored nutrient from the seed vacuoles, right? Not only we tend not to think about minerals. So we know starch becomes sugar and protein become amino acid to build the young seedlings. But in fact, minerals are also remobilized from seed vacuole and support the germination process, including potassium. Potassium is stored there, and they need to take out the potassium before the root is built up to uptake minerals from the soil. Naturally, you need all the minerals, and those minerals are from the seed storage, right? Okay, so the germination assay suggests that if CBL are important for potassium remobilization from the seed vacuole, then the mutant would not germinate very well without external supply of potassium, right? That's exactly what happened. So Y-type can germinate and grow root and seedling, not too much problem. But the mutant really can't do much. We really suffered. As you add more and more potassium to the medium, the mutant catch up, and you get to 1 millimolar or 10 millimolar potassium. The Y-type pretty much behave like Y-type in establishing a nice seedling, right? So this is a strong evidence for CBL function in potassium remobilization from seed vacuole. Now, another assay we did is with adult plant, right? Hydroponic culture plant. Under normal potassium level, they're normal, the plant. But if you transfer those plant to low potassium, 10 micromolar or 0.01 millimolar potassium in the medium, after 9 days, this is 9 or 10 days, I think, the seedlings show a very typical potassium deficiency phenotype, right? So this both assay suggests that the CBL 2 and 3 is very important for low potassium response or low potassium adaptation. Okay, so another piece of evidence, it's interesting. This is a busy slide, but all I want to say is that the mutant leaves especially have a much higher potassium, total potassium, than the Y-type. And that is very revealing, because if you think about it, if your vacuole can only accumulate but cannot remobilize the potassium out, then you keep accumulating the total potassium would be more, right? So that makes sense. So that again suggests, well, probably CBl 2 and 3 is important for potassium remobilization from the vacuole. Now, so what is downstream of CBl 2 and 3? As we know, CBl go through CIPK in order to function. So we then identified 4 CIPKs, 3, 9, 23, 26. We call it quartet CIPKs that physically interact with CBl 2 and 3 at the vacuole. This is a BC assay showing that they interact, form complex at the vacuole. Okay, so genetically, do they localize in the same pathway? So we start to mutate those CIPKs to produce a single, double, triple, and quadruple mutant, and then do seed germination assay and the hydroponic transfer assay. In both assays, as soon as you delete at least 2 CIPKs, the more you delete among these 4 CIPKs, the more dramatic, more severe the phenotype of this mutant became. Okay, so in the germination assay, the double mutant already shows some phenotype. The triple and the quadruple mutant show even more severe phenotype. In the transfer assay, right? So there's a quadruple CIPK mutant, pretty much like CBl 2 and 3 double mutant, right? So the CIPK mutant phenocopy CBl 2-3 mutant phenotype, right? Suggests that genetically they localize in the same pathway. And earlier I showed you that they form complex, physical complex at the vacuole membrane. Now, do they really regulate those remobilization process? And that process is mediated by TPK channel, right? So what we did next is to isolate vacuole, do patch clamp to directly measure the TPK activity. In the Y-type plant, you can see the TPK activity, the potassium efflux from the vacuole, right? It's a quite nice current, right? But the CBl 2-3 double mutant essentially wipe out the leak current, okay? So the instantaneous current, we often call it leak current, basically potassium leak out of the vacuole through the channel. Okay, so as you try single mutant, double mutant, and then triple mutant of CIPK, quadruple mutant, then you see the decrease of the potassium current, right? So this one, quadruple mutant of CIPK, really similar to CBl 2 and 3, essentially wipe out the potassium channel current. Okay, so genetically, this CBl, CIPK, TPK, mediated potassium efflux seem to be really in the same pathway, right? Now, can we reconstitute, go one step further to reconstitute this pathway? So we did. By cloning the CBl, CIPK, and TPK, and fuse them to different colored fluorescent protein, we can monitor the expression of a different combination of these proteins at the vacuolar membrane, and then patch clamp those vacuoles, expressing particular combination of the protein, right? So when we express a single TPK, you start to see some current, but when you add CIPK 9, then the current is much bigger. And when you have all 3 components, you have a huge activity of the channel. Okay, so this basically reconstitute the CBl, CIPK, TPK pathway, suggesting that the calcium-dependent CBl, CIPK signaling pathway activate the TPK in response to low potassium, so that potassium can be remobilized from the storage vacuole and support metabolic and cell growth. Okay, so now with that, I want to summarize what I told you by saying that, in response to low potassium, we know the calcium signal is produced through which channel is still a question mark. And with the calcium that activate two pathways, CBL19 and CIPK23 pathway get targeted to plasma membrane where they regulate, they activate AKT1 and HAK5 transporter or channel activity to enhance potassium uptake. Okay, so this can happen, you can imagine that happens in the root hair. In fact, in our original paper, we did use root hair to do patch clamp to show that this happened in root hair, okay, to enhance potassium uptake from the soil or from the environment. Now, another pathway also involves CBL2 and 3 that target 4 CIPKs to the vacuolar membrane where those CIPKs interact with and phosphorylate TPKs. I didn't show you the phosphorylation data, but they are phosphorylating TPKs. And that would activate TPK, enhancing the remobilization of potassium from the vacuole. So both enhanced uptake and remobilization work together to increase the potassium level in the cytoplasm to support metabolism. Okay, so that is what I want to tell you today. A couple of examples to show, how this CBL-CIPK network really regulate nutrient homeostasis with potassium uptake and remobilization as an example. And of course, through the years, my lab and a few other labs have identified many different transporters in plasma membrane, handling, iron, nitrate, ammonia, magnesium, sodium, potassium, of course, are all regulated by CBL-CIPK pathway. And I just show you, this TPK at the vacuolar membrane, and there are also other few pathways also regulated at the tonoplast or vacuolar membrane level. And that makes sense because the vacuole membrane, controlling storage and remobilization, and also the plasma membrane control the in and out of nutrient from the environment and the cell, of course, are two very important interface for regulating nutrient homeostasis, right? And with that, by the way, if you want to, see more details on the 20 years of research CBL-CIPK network, this is, I think, a good review. It's not because we wrote the review, but it's a most recent, more comprehensive kind of review summarizing 20 years of research in this area. Okay, so with that, here's a book talking about coding and decoding of calcium signals in plant, where your own faculty, Simon Gilroy, also contributed a chapter there. Okay, so I want to thank the people through the years, who worked on this calcium-dependent CBL-CIPK PB2C channel network. And that network, of course, regulate mostly the nutrient sensing process in plant. And the people also worked on, those coding mechanisms by identifying those channels involved in pollen tube growth, reception, and also immunity. With that, I also acknowledge NSF support and some collaborators for the years. I guess I'm ready for questions.
Great, great. Well, sorry, really impressive. It's awesome. So, questions for Sheng?
Hi, Sheng. I have questions. How are you?
Hey, Jake.
a really nice talk,
Thank
of
you.
course. And I always love the sort of big picture perspective on this. And so you had me thinking, when I read your paper showing that MLO is also a calcium transporter, it kind of blew me away because I feel like we keep on getting more and more calcium transporters in recent years.
I know.
And it's really cool. And so I'm thinking, a lot of sensing pathways, my sensing pathways, a lot of pathways people work on are sort of fine-tuning homeostasis. There's a certain range. And the CBL, CIPK pathways you're talking about are sort of in that. And then there's this other side of calcium signaling with the NLRs and MLO and other transporters that are triggering these extreme death responses, really dangerous responses.
Exactly.
And so I'm wondering how you think about, how is it that plants are able to sort of have a strict wall there to keep those two signaling pathways from talking with each other, or do they actually talk with each other on some level?
Yeah. So this is a very good question. I mean, obviously, the specificity we've been talking about, right? So this coding and decoding. I mean, I give an example, how this physical interaction really important, right? And also show you an example how this Feronia, the synergy cell, how MLO get recruited physically into the complex. So that's why we call it a trio, the kind of three proteins that they work together as a receptor channel combo in a way. So my short answer to that question is really physical interaction or recrusion of specific players into a specific place at a specific time. That really specify things, right? So the specificity is really controlled by many layers of things, right? For example, the CBL, CIPK, I mean, I often say, well, if you look at this picture, the subcellular localization can be a specifying factor, right? Specifying parameter. You localize it to plasma membrane or vacuole, or if you think about a cellular level, some cells express a particular toolkit, right? And gene expression level, cell type specificity level, all this can serve as a specifying factor as well. In terms of this, extreme calcium elevation causing death versus those moderate calcium, elevation to trigger signaling. This is I think this really emerging concept through the discovery of ANR, those immune receptors serving as a channels, kind of apparently when it's open, it's really open the hole, really.
Right.
It's open the floodgate, really, right? Yeah. So that versus a traditional channel regulation, I'll shut down, open a little bit, and then immediately close. So the several examples I give you is all have this kind of self-feedback mechanism to control the influx, right? So that it's reached a certain peak, immediately go back down, so that the total calcium level is not going through the roof, right? But in the case of ETI, I mean, those effector, receptors, it's just a very different story. It seems that the floodgate is open and then it just keep going and until cell die. So the specificity, as I said, is controlled at many levels, but definitely physical interaction is a very important part of it.
Cool, thank you.
Hi, I have a question. So you kind of briefly, in the first phenomenal talk, phenomenal studies, very impressive. And so I have a question about the involvement of ROS, the reactive oxygen species, in terms of kind of its involvement in the kind of coding or decoding of the calcium signaling. Can you speak a little bit more on that? Because I saw in your slides that ROS is involved in pathogen attack response and also kind of the potassium starvation response.
Yeah. That's a good question. I mean, the ROS and calcium always go hand in hand, as they say. They form some kind of feedback loop. It's hard to say. In many cases, it's hard to say, which one is upstream, which one is downstream. I mean, they go back and forth, and they form this cycle, feedback loop, they call it, right? In the case of immunity, many people believe calcium is probably upstream because this ROS release enzyme require phosphorylation and activation by CDPKs. I mean, that's evidence there. But in the case of low potassium response, I would say very little is known. Some studies show maybe ROS come first, but I would say the picture is still very murky there, very unclear.
Thank you.
Oh, sure.
I just wanted to say first that your reconstruction experiment on the tonoplast is really, really, really cool. But I had a question, actually more the pollen tube story where you have these oscillations of calcium. I was wondering, is it known if there is some signal encoded in the frequency of these oscillations, or is that the amplitude or the number of oscillations? What encodes this part of the signal?
Yeah, so first of all, the pollen tube growth, right? From the moment of pollination, really. I mean, I show you a pollen grain, right? Even before germination of the pollen tube, right? So the calcium oscillation already started at a particular spot. That's where the tube is going to emerge, right? And then as soon as the tube forms, then the calcium focus will be, focused at the tip of the pollen tube. And that oscillation always going on until it reaches the ovule. So throughout the pollen tube guidance, growing through the style all the way to the ovule, the whole journey, I was told, I mean, by other labs, so basically the whole journey involved this oscillation. It's just constantly oscillate. I guess this, whether there's a signal, first of all, I think there's a signal, and there's a new paper is in revision now, suggests that there should be signal, peptide-based signal that triggered this kind of calcium channel activation. But again, since then, what controlled the pace of oscillation? It's, to me, my answer is really auto-regulated, because the number of channels at a particular location is pretty much fixed after the pollen tube is there, let's say, right? And then when you have the calcium channel open, so at a particular time window, the number of calcium ion flux into the cell is fixed. And then at the peak level, the chemodulin activation or the binding association or dissociation, the K, the coefficient is also fixed, right? So reach a certain concentration, start the downturn or start the inhibition. So it's all kind of confined at a micro domain where those channels work. So it's all localized there. It depends on, I mean, the parameters is pretty much fixed if you don't care about the transcription level, right? So how transcription, I mean, it's regulated through the pollen tube growth. That's another story. I don't think anyone has work on that. But let's say if the protein number is there, then everything will be fixed.
Yeah, exactly.
It's just auto.
Yeah. Cool. All right. I think it's actually it's time. So thank you, Sheng, again for the great seminar.
Thank you, Jean-Michel, and
thanks a lot.
All pleasure. Right. Okay, bye-bye.
Thank you.
Bye.